Hello everyone, today we are going to discuss about the citric acid cycle. The citric acid cycle is a biochemical process. It occurs in the mitochondria of eukaryotes and cytosol of the prokaryotes. The question arises in our mind, why TCA cycle used? It used to generate energy like NADH2, FADH2, ATP or GTP and production of amino acid. In this cycle, there are 8 different steps. In 8 different steps, 8 different types of enzymes are regulated all these steps of the citric acid cycle. So in the first step of the citric acid cycle, there is a 4 carbon containing compound oxaloacetate is combined with acetyl coenzyme A, which is a 2 carbon containing compound is converted into citric acid, which is a 6 carbon containing compound by the presence of enzyme citrate synthase. Here 1 molecule of water is added in this process. In the next step, citric acid is converted into isocitric acid by the presence of enzyme aconitase. In the second step, there are 2 intermediate steps in between them. In the first intermediate step, the citric acid will be converted into cis-aconitic acid. Here, 1 molecule of water is released in this process. In the next intermediate step, that is the cis-aconitic acid is converted into isocitric acid. Here, 1 molecule of water is added in this process. The 2 intermediate steps are regulated by the enzyme aconitase. In the next step, the isocitric acid, which is a 6 carbon containing compound, is converted into alpha ketoglutarate acid, which is a 5 carbon containing compound by the presence of enzyme isocitrate dehydrogenase. Here, isocitric acid is a 6 carbon containing compound and alpha ketoglutarate acid is a 5 carbon containing compound. So, in 6 carbon containing compound, there is a 1 carbon is removed by the process of CO2, and in this process there is 1 molecule of NADH2 is removed. In the next step, the alpha ketoglutarate acid, which is a 5 carbon containing compound, is converted into succinyl coenzyme A, which is a 4 carbon containing compound by the presence of enzyme alpha ketoglutarate dehydrogenase. Here is a 5 carbon containing compound is converted into 4 carbon containing compound. There is 1 carbon is removed in the form of CO2. Here another 1 NADH2 molecule is released in this process. In the next step, the succinyl coenzyme A, which is a 4 carbon containing compound, is converted into succinate by the presence of succinate thiokinase. Here 1 molecule of GTP or ATP molecule is produced. In the next step, the succinate is converted into fumarate by the presence of enzyme succinate dehydrogenase. Here 1 molecule of FADH is generated. In the next step, the fumarate is converted into malate by the presence of fumarase. In the last steps of the cycle, the malate is converted into oxaloacetate by the presence of enzyme malate dehydrogenase. Here another 1 molecule of NADH is released. One thing we should remember that in all these 8 steps, what are the reversible and what are the irreversible steps? In step 1, step 3 and step 4, these 3 steps are irreversible in nature and remaining all 5 steps are reversible in nature. Hopefully you understand all these concepts related to the citric acid cycle. Thank you.